everyone. Thanks for coming. Before we start, I'm going to read our land acknowledgement. Berkeley Public Library buildings are built on the territory of the Huichin within the homeland of the Chochenyo-speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley and throughout the East Bay since time immemorial. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unceded stolen land, and we pledge to recognize the permanence and the rich contributions the Ohlone people continue to bring to the city of Berkeley and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. The Berkeley Public Library is committed to helping create a more just and anti-racist society and to furthering the visibility and celebration of Ohlone authors who are specific to the East Bay and Ohlone culture in our collections and programming. This is a living document that was co-written by Ohlone cultural leaders Vincent Medina and Louis Trevino and the Berkeley Public Library. Welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have two speakers at each seminar who will talk about their current research or a topic they find really interesting. The organizers are two graduate students at UC Berkeley, Kaylin Zhuang and Daniel Bretthauer. This program happens every third Tuesday of the month at 5:30 p.m. at the library and on Zoom. If you're interested in checking out their past seminars, you can visit their website and sign up for their listserv to keep in the loop for upcoming seminars. Also check out their YouTube page to view this lecture and see an archive of past lectures. Thank you so much.
Thank you very much, Molly, and welcome everyone. Thank you so much for coming, and also thank you to our speakers, Lauren and Makenzie, for volunteering to speak with us tonight. And like Molly said, this is a open format talk, so please feel free to raise your hand throughout. And for folks on Zoom, please feel free to use the Q&A button or channel, and we can ask the questions online. And without further ado, it's my pleasure to introduce the first speaker of the night, Makenzie Wilkinson. Makenzie grew up in a small town called Hollidaysburg, Pennsylvania, with her mom and brother. And she shared that don't let the cheerful name fool you, because it was -4 degrees outside during the major East Coast ice storm these last few weeks. Makenzie did her undergraduate at Johns Hopkins University, where she pursued a degree in neuroscience, and before working for a few years with children diagnosed with severe developmental disabilities. After the peak of the pandemic, she re-entered the academic space by completing a postdoctorate program, research program in the lab of her current mentor before entering her PhD in neuroscience department at UC Berkeley, beginning in 2023. She joined the Dolan Lab as a graduate student and have been studying multiple types of psychedelic drugs and their potential to reinstate juvenile-like plasticity in adult brains. Their current approach uses both mice and octopuses, which offers a unique way to study how these substances work across different brain types. Outside of research, she can usually be found foraging for mushrooms or out camping with her friends, and outside of that, she likes to read fun fantasy books and spending time with her two cats, Pandora and Narcissa.
Thank you. I just want to start by saying thank you so much for the invitation to be here. I am very excited to take everybody on a really fun trip to get into what my lab, that will not be the first joke of the night, to get into what my lab studies, which are how psychedelic drugs reopen critical periods of development. So to start out, when I was younger, my mom used to keep a Far Side comic book gallery in our bathroom, and I used to love flipping through these as kids. And even now as an adult, I still find a lot of enjoyment for some of the really funny comics. This is one such example, and hopefully by the end of this talk, you'll understand why this is one of my favorites. But to get some crowd engagement, first, I'm just curious if anybody knows what this is in reference to. So for anyone who can't see, the caption is saying, when imprinting studies go awry, and its description is this scientist who seems to be following behind a mother duck as if he's one of the baby ducklings. Has anybody heard of this or like familiar kind of with what this is referencing? Yeah, awesome. So this was Gary Larson's take on an observation made in the 1940s by an Austrian zoologist and ethologist known as Konrad Lorenz. You've probably seen pictures of Konrad Lorenz looking like this, or perhaps this is a really famous one. But he basically observed that within the first 48 hours of a gray-lag goose's life, there was this bond that would form with the very first moving object that that animal saw. Now, this was extremely time dependent, so it had to occur within the first 48 hours of hatching that a goose would form this really lasting bond with the first moving thing that it saw. Sometimes it would be the mother goose. Oftentimes it would be Konrad Lorenz himself. He kind of noted this window of time where the brain had the ability to form this lifelong bond as a critical period. And we can kind of understand these as periods with which the brain is expressly sensitive and malleable, so able to make changes given information that's really relevant to them in their environmental space. I think a really easy analogy to describe critical periods would be the wet brain in cement, in which we can think that when the cement is wet, we would be able to redraw and change and restructure the brain, which whatever way we see fit. And then when that cement dries, all of those changes are going to become permanently like locked in place. Now, this is kind of reference to, or gives reference to another common topic that maybe people are familiar with, known as neuroplasticity. Does anybody want to give me like a basic definition, or maybe throw some ideas as to like what neuroplasticity really is? You can feel free to shout them out.
Like how the brain can kind of reassign those to different parts, like one part is damaged.
Yeah, absolutely. That's a perfect and like probably the most well-known example of this. So neuroplasticity at its most basic definition is the brain's ability to kind of change and learn. So critical periods and neuroplasticity go really hand in hand. So early on in development, when you have open critical periods, you have a lot of neuroplasticity, a lot of the ability for this brain to kind of relearn things and recircuit itself in whatever way it sees fit. And over the course of development, critical periods close, and we also see a massive loss in this ability to make changes in the brain. Now, as it turns out, critical periods didn't just apply to gray-lag geese. They're found kind of throughout the entire animal kingdom. We have critical periods for song learning in zebra finches and other songbirds. And you have critical periods for organizing the brain areas responsible for the whiskers of a mouse. As human beings, we also have critical periods, as I'm sure that we can imagine. We have critical periods for vision. So when we first open our eyes, when we're first born, there's a period with which the brain learns what information is coming from which eye, and it keeps that organized in our visual centers. We have critical periods for hearing and audition, because in the inner ear, we need to map correctly where different frequencies are and then have those frequencies correlate to other areas of the brain so that we're sure how we can process audible information. And then maybe one of the most well-known critical periods is that for language. So if you've heard the idea that it's way harder to learn a language when you're an adult versus when you're a kid, this kind of gets into the same idea. And really, one of the most fascinating topics for me when I was learning neuroscience was for the critical language, or the critical period for speech, which is that you only have the first 7 years of your life to be able to develop speech or to be exposed to speech. And if you pass that point without any exposure to it, you will never regain the ability to learn how to speak. We even have some critical periods for post-motor learning that happens after you have a stroke. So brief windows of time when you're able to try and regain some of the abilities that perhaps were lost. Now, critical periods close, and that's a good thing. It might be really energetically costly for our brain to try and maintain this high state of plasticity. And you can imagine that you don't want to wake up every day and have to relearn how to see or how to hear, or imagine that some new information overwrites the language that you speak. That would be really hard for us to do. So critical periods close. It's a good thing that they close, and it gives us this ability to gain as many skills as we can in the time with which they're open. When they close down, then those skills get locked in place and we don't have to worry about losing them. Now, despite me just describing why it's a great thing that these critical periods close, can anybody think of any ideas or any problems that might arise if that critical period stays closed, or maybe a reason that we might want to reopen them later on? Any ideas? Yeah.
Something gets damaged, you still want to have plasticity to be able to regain it or something?
Absolutely.
Or whatever.
Perfect. That's my first example. So in response to injury, so imagine something like you get a traumatic brain injury and you lose a lot of functions. You would want to have the ability to reinstate some of those lost functions. We can also think about if you experience some type of traumatic event. We think PTSD, and now you've adopted some type of maladaptive behavior that's not necessarily beneficial to you or really significantly impacts your day-to-day functioning. We also, oftentimes in adult populations, have a way better chance of diagnosing different neuropsychiatric diseases. So when we see treatment-resistant depression, OCD, intense anxiety, a lot of these things happen later in life, And the problem is we might have all of the tools to go in and try and fix those changes. But if the brain is in a state where it can't necessarily change itself, then we're not going to be able to do all that much good. So our lab has this idea that it would be really beneficial if we had some type of key later in life that would be able to reopen these critical periods, even for just a brief amount of time, so we can go in with the therapies that are needed or the medicines that are needed in a state with which the brain can actually take on those changes. And as it turns out, we found that key, and those keys are psychedelic drugs. So we've seen an upsurgence of recent successes with different clinical trials regarding psychedelic drugs, those for depression, for PTSD, for substance use disorders, including opioid dependence, and even treatment-resistant major depressive disorder. Now, the results for these are extremely promising. We're seeing people do one treatment and have lifelong or months and months and months where they no longer have the same symptoms. But nobody has identified a specific mechanism that describes why this is happening, especially because across all of the different drugs that are used, they don't necessarily work the same way in the brain. So for example, a lot of these drugs don't bind to the same receptors. So it's really hard for us to track down, well, what's then responsible for why they have such an incredible ability to heal adult brains. In order to begin answering some of these questions, my lab decided to look at, okay, well, let's look across different psychedelics that maybe aren't necessarily all within the same class to start to see what's changing in the brain, especially in adult populations that might be underlying this really incredible therapeutic potential. So these are the different psychedelics that we use. I'm sure there are many up here that a lot of people recognize, but is there anything on this list that somebody's maybe never seen before, or a psychedelic that seems unfamiliar to them? Ibogaine, perfect. That's becoming a lot more recent, but it's definitely probably the most the least well-known out of all of these different psychedelic drugs. This comes from the root of the iboga bush, which is native to Western and Central Africa, and it has really intense psychedelic effects. The trip itself lasts anywhere from 36 to 72 hours, so it's an intense one. Exactly. Ibogaine is pretty intense, and so I've kind of introduced to you these two different ideas, one of which is that we have these critical periods. These critical periods close, and it'd be really beneficial for us to reopen them for therapeutic benefit. And we also have this idea that there are these psychedelic drugs which seem to be having an incredible therapeutic benefit in adult populations, but we can't figure out exactly what mechanism describes that. Well, my lab had the idea to test the hypothesis that maybe the reason psychedelic drugs are so helpful in adult populations is because they're reopening critical periods of development that typically close early on in life. Now, in order to study this, we're going to need some help from some of our friends. And unfortunately, I'm not going to be sharing a lot of the octopus work that we do in our lab today, but I'm very happy to talk about it if anybody has any questions. I'll include my email, and you can just ask me throughout the talk. But now we're going to enter my mouse friend, which is how we do most of the psychedelic studies in the first place. In order to test whether a psychedelic can reopen a critical period, first we need a critical period for us to be able to reopen. So my lab found that there was this critical period, a novel one for social reward learning in mice, and we can study that using this basic paradigm here. And I'm going to walk us through it really slowly. Basically, a mouse will start in its home cage on a bedding that it's really familiar with, and it's typically with its same-sex siblings that kind of it grew up its entire life with. So really stable social history. We then take a mouse and we put it into what we call a preconditioning trial, in which all we're doing is exposing the mouse to two types of bedding that it's never seen before. So it spends 30 minutes just freely exploring this cage, just getting exposed to bedding A, which we're going to call the blue bedding, and bedding B, which we're going to call the tan or the yellow bedding. After 30 minutes of just initial exposure, there's nothing that they need to be doing in this time. We're then going to start to put them into a context learning training situation in which for 24 hours, a mouse is going to move to this social condition in which it's placed back with all of its other cage mates on one of the beddings that it had just been introduced to. And over the span of 24 hours, we want this mouse to start associating blue bedding. Bedding A means I'm with my friends, I'm with other mice. After 24 hours, we move from this social context into an isolation context where, as you probably guess, it's just learning the opposite context. So now we take our bedding B, our tan bedding, and we're teaching the mouse that when you're on tan bedding, it just means that you're by yourself. After it's formed this association, we put it back into this box that has kind of both of the beddings on either side. And we're simply just seeing, does it spend more time on one of these two sides once it knows what those sides mean? Over the course of development, so starting really early on in a mouse's life, testing all the way up through when it's a mature adult, we see a really intense developmental profile of this type of behavior. And this just means that when a mouse is really young, it spends way more time on the social betting side than when it's an adult. And it's not necessarily that the adults don't want to be around other mice. They're social in much the same way that we are. They just don't really care whether they were alone or whether they were with other mice. And now because this critical period curve that we're seeing was so robust, we started thinking, wow, like we may have maybe found a critical period that also has some implications for human diseases. I mean, I think we can all think back to when you're in middle school and it's really important to you what your friends are doing, what things they like, or what clothing that they're wearing. And the older you get, the less you really care about some of those things. So we found this critical period and we found a behavior for this critical period, but we wanted to take it one step further. And I'm not necessarily going to get into really specific details about this technique, but I think it's a really awesome opportunity to introduce to you some of the ways that we can not only study the behavior a mouse has, but also what's going on in the brain at the time that those changes are happening. So early on in life, as we just described, a mouse is exposed to other mice and it all of a sudden really, really, really wants to be around other mice. And we can study this by showing that I take this really, really thin glass electrode, so just a really sharp needle, and I can get down to the level of an individual neuron in a mouse's brain and poke it. And once I poke that neuron, I create kind of a free fluid between the inside of the neuron and the inside of my electrode. And that just allows me to record the signals that are both coming into that neuron and that neuron is sending off. And so I have this ability to measure, well, what changes are happening in the brain at the time when we say this critical period is open versus the time when we say that this critical period is closed. And early on in life, when you're exposed to other mice, you get the release of a drug called oxytocin. And people might be familiar with oxytocin as what we call the love hormone. So it's released when you have a baby, when you fall in love. And one of the maybe lesser known roles that oxytocin has is that it is released when you go through a social situation. So just when you're around your friends, oxytocin gets released. And so in a juvenile state, when oxytocin is released, it causes a very specific type of plasticity that I can measure using this technique of electrophysiology. When the mice are adults, the same oxytocin is released in a social situation, so they also get oxytocin, but all of a sudden, we no longer see that same plasticity happening. So we have two things going on now. We have a behavior that we can measure really easily, in which when mice are young, they really choose to be around other mice. And I also have this thing that I can do inside the brain to measure the plasticity that's happening when they're in that open state and when they're young, and watch that plasticity go away when they're in an adult stage. Again, if anybody has any questions, feel free to interrupt me at any points of this. Yeah.
Does this have to do with like the novelty of things, like like the brain craves like new shit, and once you've seen a lot of stuff, I know that was not science jargon there, but once you've seen the stuff, you're just like, oh, whatever. So it's a newspaper. It says the same thing.
It can get into the same ideas. What we're really looking here is for the reward value of those social interactions. So there are tests where we can see, does a mouse prefer to be around other mice, or does it prefer just a completely novel object? And sometimes they do. A lot of times when the mice are juveniles, they really crave other mice, but that like overdevelopment, it doesn't really change whether or not they choose the novel object. They're always interested in something novel when they're older. But in terms of just interacting with like a social environment, they really, really care about social when they're young. They don't really care about social all that much when they're older. So I brought together two ideas. Remember, we have psychedelics. We want to try and reopen this critical period in the adult brain. And we have these critical periods that need to be readjusted. And so our basic workflow is going to be that I take a mouse and I give it a drug. Sometimes that's a psychedelic. Sometimes that's just saline, which is just a very basic control to make sure that nothing should be happening or nothing should be changing with saline. After the mouse experiences the full trip, the whole time that it's under the effects of the psychedelic drug, I then put it back into its home cage and I just let it sit there. Now, this is really important because our lab isn't as interested with what actually is happening during the trip itself. That's cool, and there's a lot of people that are looking into that, but we're really interested in what's changing in the brain afterwards that's allowing for some other changes to happen. After this, the mouse is either going to go into the same behavior that we just talked about, or it's going to be tested for electrophysiology. And all I'm looking at is, if I give a psychedelic drug, does it behave the same way that a juvenile mouse behaves? And the first one that we tested was MDMA. MDMA we call an empathogen. Maybe some people recognize this cuddle puddle here at the bottom, but MDMA is a drug that makes you really want to be social. You really want to be around your friends. You want to be around others. So we thought, we're trying to reopen this critical period for social reward learning. Maybe a psychedelic drug like MDMA is going to be the best one because it itself is also inherently social. And what we can see is that, again, thinking about what this critical period looks like, when we just give it saline, it behaves the same way as every other mouse behaves when they're an adult. It just doesn't really care. It has no strong preference. But when we gave it MDMA, all of a sudden we get this reinstatement of the same way that the mouse behaved when it was a juvenile. And that seemed really interesting and like a really cool finding. But again, maybe not super surprising because MDMA is itself inherently also a social drug. So we tested it out with others. We then tried psilocybin. Psilocybin's not really social. Maybe you like being around other people when you're on psilocybin, but it's nothing about the drug that really makes you crave that social interaction. Instead, it's more of what we consider hallucinogen. So a lot of like sensory and visual experiences are happening. Well, it turns out that when we gave this mouse psilocybin, it also behaved the same way as when it was a juvenile. And both of these things are also happening in terms of the changes in the actual brain. I'm just showing you the behavior data because it's a little bit easier to see. So we tested psilocybin and it worked. And we tested LSD and it did the exact same thing. Now, when we give LSD, all of a sudden our adult mice are behaving the same way that they were when they were juveniles. And those ketamine and those ibogaine, again, different drugs, you have different experiences on them, but they all seem to be doing the same thing, where an adult mouse is now behaving and its brain is behaving as if it's a juvenile. This was a really incredible finding and a really shocking, surprising finding for our lab because we weren't expecting anything beyond MDMA to work. But this does seem to tell us some type of universal truth about the way that psychedelic drugs are changing the brain over the course of time, so that when maybe you have gotten some type of neuropsychiatric illness or you yourself have experienced PTSD, when you go through a psychedelic clinical trial, it's not necessarily that the psychedelic drug specifically is doing something to go in and fix all of these problems, but instead it's kind of opening up this window to learn. And then when you go through the therapy, when you start to ingrain those changes, then maybe that's what's explaining why we see people having this really incredible benefit with psychedelics way after the fact. Another really shocking thing for us was that the amount of time that the mouse spent in the psychedelic trip, so let's take something like ketamine, really short acting, that time was directly correlated with how long the critical period stayed open. So our short acting drugs like ketamine, that critical period only stayed open for like 4 days or so. But our long-lasting drugs, let's take LSD, which lasts from like 8 to 10 hours, that critical period stayed open for a span of like 3 weeks. And ibogaine, the one I told us about at the very beginning, the really, really intense psychedelic drug, that one stayed open for at least 4 weeks. And that's as far out as we tested it. Could have stayed open for even longer. This type of information was really useful to us because we might think about how we can inform development of different clinical trials or what things psychedelics might be useful to treat. And so I brought up that we had a critical period for stroke. And that right after you have a stroke, there's this brief window of time that your brain allows you to try and regain other motor functions. Well, that critical period closes pretty quickly. And that's why you see a lot of people who have lifelong impairments, whether to their speech or their motor abilities after experiencing something like stroke. But maybe this result is helpful for us because if we need to open a critical period for even longer, perhaps using a longer lasting psychedelic like LSD might be useful for clinical trials with stroke because you need a lot of time to regain the ability to speak or the ability to walk. And maybe the shorter acting psychedelics like the psilocybin or the ketamine are really helpful for things like depression or anxiety disorders where you want to get in and you want to have time to make those changes and do your therapy. But a lot of times people are going back to the same environments that were maybe initially problematic or traumatizing to them. And you don't want to leave people in a completely open state where anything could re-traumatize them or they could learn bad behaviors again. So a shorter acting psychedelic might be more useful here. And informing this decision was these mouse studies, but also, as I told you, we do some work with the octopus. And again, I'm really happy to get into that at the end. But in terms of future directions as to where we're going, you don't have to pay anything on this chart. And I'm happy to provide this resource. They said they're going to post this on YouTube after this. But this kind of is showing us that it does seem to be the fact that when people get a psychedelic drug and they also get therapy at the time they're going through the psychedelic, their results last for way longer than people who have a psychedelic experience, but there's no therapy associated with it. And maybe they also still are seeing like symptom reduction for things like PTSD or for depression or for addiction. But there's something inherent to the mechanism that the cell somehow knows when it's in one context, when it's getting therapy and when it's not getting therapy. And that's a really difficult problem to solve. And so most of my thesis work is going to be based on how can we ask these questions in mouse models where it's really difficult for me to ask a mouse, like, how was your trip? How did you have a good time? How can we get into questions of context and set and setting that describe why in the human populations we're seeing way longer lasting psychedelic results from the psychedelic clinical trials? And I'm happy to speak a little bit more about that. And the last thing I'm going to bring up, just because I don't want to take too much more time, is just that, as always, the same that we did this land acknowledgement, I love bringing up the fact that psychedelic drugs are not new medicines. We've known about them for millennia. People have been using them for millennia. So I like to think about the work that my lab does and the work that we do as being informing of truths that we've probably known for a really long time. And just because there's maybe not empirical scientific evidence behind something doesn't mean that it wasn't true and it wasn't helpful, and especially helpful for larger populations of people from beforehand. So right here, I just have two different practices with another psychedelic. This one is ayahuasca, in which people to this day are still doing ayahuasca. Ayahuasca tribes and clinics are still up and running. And this is one of the most ancient forms of, or one of the most ancient pieces of evidence we have for ancient like hallucinogenic use. And it's a pouch that used to keep ayahuasca that's over 1,000 years old that was uncovered. So just kind of making the point that these things have been around for a really long time. They're really useful and really beneficial and incredible as a thing to study as a scientist, but not necessarily something that we've just completely discovered within the last 10 or 15 years. And so with that, I'd also like to thank all of the people that went into doing this research, the lab members that I have, both current and previous, who helped us to publish some of these papers. Here are some fun pictures of my lab with people who have come to visit. This is on Halloween, where my lab and I dressed up as witches and we went to an apothecary. And if you're interested at all, I have my email here, our Dolan Lab website. You're also able to find our lab's phone number where you can call and ask questions or just get into contact with any of us. And with that, I'd like to say thank you again, and then please let me know if you have any questions.
Urban Place is your lab?
No, this is just a picture that we took in front of the lab. Yeah, no. This is just by the lab, but that's a picture of my PI, who's kind of the foundation of all of this. Yeah, question.
Psychedelics, but what is it that make all of those drugs we talked about psychedelics? Like, what do they share chemically, or how they act in the brain?
Yeah, absolutely. I'd say that there's subjective experiences that kind of cohere the category of psychedelics. And so a change in your perception of time, of space, of your visual acuity, things that are changing with terms of like the things you're experiencing under the psychedelics, they all seem to be having this effect of a metaplastic change, meaning that they're not necessarily just causing a wide burst of plasticity, but really they're opening up these windows for plasticity. And the way that we can start to differentiate that is to test them against drugs like cocaine. And so cocaine is a really perfect example of a drug that's also psychoactive, that also causes an altered state, but it doesn't necessarily do the same thing that these psychedelics do. And then beyond that, we try to go back to literature where people who are originally making psychedelic drugs, how they describe the different chemical classes and what things are shared between them, and what results we're seeing in terms of clinical trials now that have the same type of one-time or two-time use with a long-term benefit towards the population that it's trying to help. Yeah.
So you mentioned that adult mice also release oxytocin during social behavior.
Mm-hmm.
I'm wondering if you additionally inject oxytocin, do they also may behave more like socially active compared to just the amount that they release?
We don't do that in terms of the actual behavior, but for the electrophysiology that I study, what I'm doing is bath applying. So I just kind of dump oxytocin onto the slice and then see how it reacts. So that would maybe get at the question of being like an artificial intense amount of oxytocin that we're getting. And even in the adult brains, if they haven't gotten a psychedelic, that dump of oxytocin doesn't do anything to cause the plasticity change that we see when they're adults, but when they're juveniles. But if we gave them psychedelics 48 hours in advance, then I dump all of that oxytocin on, all of a sudden that plasticity change comes back. Yeah.
Have you had a chance to look at the effects on like other critical units, and what do you find?
That's a perfect question, and exactly what this research kind of led to is the context that you're in when you're doing the psychedelic trip, is that what directs which critical period opens? And so we have really preliminary evidence with some of our collaborators back at Johns Hopkins, and they're looking to see if you give a mouse a psychedelic under a motor context, will it reopen the motor critical period to help with something like post-stroke? And one of the things I'm interested in is looking at that context dependence. And so one thing I didn't share was that when we gave MDMA to the mice, it would only reopen the critical period if MDMA was given in a social setting. And we thought that, okay, maybe there's something about MDMA because it's just a social drug that requires. But when all of the other psychedelics also reopen the social reward learning critical period, one of the first things that I'm going to test now that we're here at Berkeley is if I do that context dependence, if I give the psychedelic in isolation versus if I give it in social, will only the ones that the psychedelic was given in social open the social critical period? And then how else might we be able to kind of broaden the scope of this to critical periods like those for language or like those for the visual critical period? There are actually some groups that have looked at psychedelics for the visual critical period and also see the same type of reopening of the parts of the brain that are specifically associated with like learning what information is coming from which eye.
Is there any reason that it is opening specific critical periods instead of just kind of like reopening all of them at once?
Yeah, so it would be a really big problem for us if it reopened every single critical period, and you'd probably see some type of long-term changes or effects that were happening, like looking at the parts of the brain that are associated with vision or and seeing how those maybe rechanged or remapped themselves. But that's an excellent question and something that I think is informative of why we think psychedelics are helpful, but also something we try and keep in mind because it's not a good thing to just cause widespread crazy plasticity in the brain. That's how drugs of abuse work. That's how addictive drugs typically have their properties. And so we call this the melty brain problem, in that it's really odd to us that you have something that might work as this master key for kind of unlocking critical periods, but it doesn't cause an all around meltdown in the brain. And what really explains why that's not happening and why they just seem to be good for certain critical periods that are directed by what you're experiencing during the time of the trip.
I was wondering, are there any other like classes of drugs that people are looking at to induce neuroplasticity besides psychedelics?
Yeah, so in terms of neuroplasticity, there's definitely a bunch, and this gets into a lot of the debate between why, whether or not it's really helpful that psychedelics cause like a lot of plasticity. There's kind of a debate in the field as to the way people think about this. Some people are saying that, oh, you give a psychedelic, all of a sudden we see a bunch of new neurons, they're all growing, they're all going in crazy directions, and that's great and that's awesome. But that's, like I said before, how drugs like cocaine work or heroin. This is the basis of a lot of the addictive drugs that we see. And so we are looking to make sure that like the plasticity that we're seeing is specific to this class of psychedelics that don't seem to be super addictive, that don't seem to have the many negative side effects that other drugs that might be causing the same type of plasticity we were originally thinking psychedelics work under. That's like a good way for us to separate those to make sure that the psychedelics stay safe, because for the most part, they are pretty safe. Not that they're not important and powerful agents of like changing and making you become a lot more suggestible. Like there are just neutral tools, but it's important for us to make sure that like they stay safe in the way that we've understood them to be. So they're not really addictive. There's not that much of a an effect in terms of like a negative sense that they're having on your brain the way that we originally thought that they were. Yeah.
Can I just say something about the like when people think about it, the addictive drugs? So 95% of people that show signs of addiction have 3 or more categories of childhood abuse. So like there's direct correlation. It's not like the drug causes people to be addicts or anything like that. Things that are in their life cause them to be out of balance and use things out of balance, as opposed to, oh, well, cocaine is a drug that makes people addicts and, you know, DMT isn't or whatever.
Yeah. I think that this gets into a really excellent point that our lab thinks about a lot, which is the difference between what we call the biochemical imbalance model, which is where you take something like depression and say, okay, depression is because we have a loss of serotonin. And so the way that you fix that is you just fix the serotonin. And maybe that's a little bit of a solution, but as we've seen, like SSRIs aren't incredible for totally curing somebody of their depression, and they come with a lot of other negative side effects. We also can think about these things as a learning model, which is that most often there is some type of root cause, and it can be an experience that you had when you were a child. It can be something that's maladaptive in the way that your brain learns information that leads to a lot of later on neuropsychiatric illnesses and diseases. And if you're not solving the root problem of something, you're probably never going to really get at a full and total cure, if there is one, for some of these different diseases and disorders. So the way that we think about psychedelic drugs follows what we call this learning model, which is just they're opening this window for you to learn anything that you need to learn. It could be good, it could be bad. It's not necessarily that the drug itself is this wonder pill that just cures your PTSD as soon as you take it. We've seen people who take recreational use of psychedelics and they're not just totally absolved of their symptoms. But thinking about it in terms of the biochemical imbalance model, you would maybe be directing your studies to just saying, oh, psychedelics are kind of a wonder pill where you take it and they just give you more serotonin, and that's why they work for depression. Or you take it and they kind of take away some of these like bad connections that your brain has made in terms of addictive drugs. And that's maybe not super helpful for us because it seems like they have more widespread and more like a better way to understand them is to think about them in terms of like what learning do they allow to happen, and then how does that change what goes on in terms of like your long-term effects from being absolved of like PTSD or major depressive disorder or OCD, things like that.
Does it, so like is it ERMMD or whatever the
the therapy.
For whatever reason it causes you to see your trauma differently, or for you to be able to understand that it's in the past or something like that. When you're using the psychedelics and stuff like that, like is it similar, or do they work together somehow, or like do people use both of them?
Yeah, I think that EDMR, I believe is what it's called, is another form of therapy that some people are using. And I think they're working on maybe similar topics. And that when you're going through a psychedelic experience, for us, it's really important that you have the actual psychedelic experience, because a lot of what's happening is the perspective change or the shifting of, okay, well, why do I feel this way about this certain topic? Or maybe there was something from my childhood or my past that I wasn't really considering that is really actually affecting how I'm behaving as an adult now. And I think that something like EDMR, I've heard, has very similar, like the route of administration and the way that people think about the way it's working in the brain is very similar. So they could definitely be linked. I haven't looked specifically into EDMR, so I'm not as familiar with that. But in terms of psychedelics, we think it's definitely something to do with like the experience of having your kind of perspective shifting on some of these other, maybe other things. It is. Yeah.
Oh, no, I'm really curious what like critical periods there are in octopuses and what you found. Like, it's crazy that something would work on both octopuses and like mammals.
Absolutely. So one of the first things that we did was even just seeing like, do the psychedelic drugs work in the same way in an octopus? And so an octopus is a really asocial species. So most species of octopus, they will kill each other on sight. Like they don't like being around other octopuses. They'll eat each other immediately. And so we wanted to see, yeah, they're extremely asocial. And so we wanted to see, well, a drug like MDMA makes us social. It seems to make mice kind of social. Does MDMA make an octopus more social, so much so that it could interact with another octopus and not immediately want to kill it? And so we did an experiment where we gave one octopus MDMA, and then we put it in a tank where it had the ability to explore just like an empty cage, or that cage maybe had a novel object in it, or there was another octopus. And if you don't give them the MDMA, the two octopuses, it'll immediately go and try and attack the other octopus, or they stay very far away from each other with all of their tentacles kind of condensed in together. But as soon as we gave the octopus MDMA, its tentacles started floating out, and we call this the ballerina pose, where it was laying over the other octopus. It was putting its tentacle in to like interact with it. And so that seems like, oh, okay, well, yeah, MDMA makes everything social, so cool. But an octopus's brain is so maximally different from ours. We didn't even recognize that maybe they had the same receptors that MDMA would even work on. So the fact that the MDMA worked to make them social tells us something really important about the evolutionary history of sociality. Like maybe it's not necessarily that you need a brain area like the amygdala, which processes our emotions, in order to be social, or you need a centralized brain in order to be a social creature. There's something that seems to be shared with something that split off from us 650 million years ago in terms of their evolutionary history. And for some reason, it still works here. And so once we did that experiment, we decided to continuously test, well, maybe this idea that like there is a social window in an octopus can be manipulated with psychedelics, the same way that this social window seems to be able to be manipulated with mice. And we're really early on in doing those pilot experiments. We just got access to our octopuses here at Berkeley. And so we're definitely going to be doing a lot more experiments to kind of test some of those ideas, and then also see maybe if it has a critical period for something like arm regeneration, or like some of these other things that are really intrinsic to an octopus and what makes it such a cool species. Like maybe there's something that we can do here with manipulating that in terms of the different psychedelics we're giving it. And then what truths can we draw from that about what things apply to us, what things apply to the mouse, and what things are true over like evolutionary history, depending on all the different brains that we're observing. Do you have a follow-up?
Yeah. Did you give both of the octopuses MDMA, or like just one?
Just the one. So the one that was inside of the tank was still freaked out. He didn't love that the other octopus was up and next to it, but he was in a little cage, and so he wasn't going to attack it or hurt it in any way. But the other octopus that was just freely in the cage, he was having a grand time floating around and kind of interacting.
Is that don't normally have?
It didn't seem to be anything specific to do with mating. We characterize it as what we call the ballerina pose because you don't really see it other than when an octopus is just totally by itself and kind of just like exploring in its environment. And so characterizing that and like looking into more details is something that we're actively pursuing in the lab, but we don't have like a ton of specific answers right now. To interact with another octopus like that. I'm sure that they do something similar in the wild, but I don't know if it's been characterized as that ballerina pose before. But I think that they do. It seemed to be a pose that signified that the octopus was just comfortable, that it was like really feeling loose and like excited to kind of be in the environment and interacting with its environment. Yeah.
How are you measuring plasticity via whole cell recording? What is the physical change?
Absolutely. So we can go back for just a second. The plasticity that I'm measuring is whether or not the signals being sent to the cell that I'm recording from are starting to decrease. And so the way that I do that is, as I said before, I've patched onto the cell, and the cell allows me to measure how many signals this cell is receiving and how many it's sending out. And when I put oxytocin in the bath, when it's in either the juvenile state or just after a mouse has gotten a psychedelic, what I see is a really stark and statistically significant reduction in the amount of signal that's coming to this cell. And when it's an adult mouse and it's in the saline condition, I still see the same, basically a high level of frequency of, like a high frequency of inputs coming from the cell attached to the cell to this cell. And so basically just measuring that really simple change of whether or not there's more signal coming or less signal coming can tell me a little bit more about what's going on in this larger circuit that we see. And the circuit is a little bit more complicated because it's kind of dependent upon oxytocin gets released, and that causes a release of serotonin, and the serotonin binds to serotonin receptors, and those might prevent the release of another signal from a cell. And so it gets complicated within the circuit itself. But even just being able to tell the like very basic change of this one means that there's high frequency stimulation and this one means that there's low frequency stimulation, and looking at how that correlates to our developmental trajectory over here tells us something about the plasticity that's happening in the brain at those different time points.
And then I also asked, how do you measure how long the critical period stays open?
We basically just keep retesting the mouse. Yeah. And so for something like LSD, which was a longer lasting psychedelic, we would just take then the mouse and put it through the social behavior again and see, does it still behave the way that the juvenile behaves? And at different time points after that initial test. So let's say we gave a bunch of mice LSD and then we waited for 48 hours. And some of them we tested them 3 days later, and some of them we tested them a week later, and some we tested 3 weeks later. And we can kind of go through both the ephys and the behavior and just see, is it still behaving the way that we expect it to? And once we see that it stops behaving, then that tells us, okay, then this is where the critical period is closed after being opened with this specific psychedelic, and kind of shows us that also really exciting result of the amount of time, for some reason, that the mouse spends tripping is directly correlated with how long that critical period stays open. And there's some molecular mechanisms that I'm starting to test in my PhD work that describes, well, why is that? Why is it that a short amount of time spent tripping leads to a critical period only being open for a short amount of time versus a long amount of time tripping leads to a long critical period being open?
What about microdosing?
Excellent question. We've started some really early experiments into testing something like microdosing to see if it has the effect of fully opening a critical period, or it might just be beneficial for less known reasons. And we didn't see, when I gave like a smaller dose of psilocybin and a smaller dose of ketamine, it didn't seem like it had the same effect of the full dose. So you had to go through like the experience for it to be tripping, for the critical period to be opening. But it does call into question the chart of this chart that we made, which basically is saying that like there are times with which I think that the psychedelics are beneficial. Going through the psychedelic experience is beneficial kind of no matter what, but it just seems like maybe microdosing is something that would be beneficial in maybe more shorter lasting time points versus the full trip might be more beneficial for like keeping the critical periods open for a long amount of time. Yeah.
A little bit of a follow-up. So how important is the sort of the role of consciousness, I guess, being awake during that experience? Like if you give somebody psychedelics when they're unconscious, does it have a similar effect?
Absolutely. That's actually a study that was done. And so for right here, we were testing depression. And in the ketamine ones, you can see one group gave ketamine and then immediately gave it, ketamine also is an anesthetic. So they gave ketamine right before somebody was about to go through surgery, and they wanted to see will their depressive symptoms resolve after they have gone through the surgery, but without having experienced the ketamine trip. And we compared it to somebody who got ketamine, but they experienced the ketamine trip. And the people, is this touchscreen? There we go. In the groups in which ketamine was given under anesthesia, they didn't really see any therapeutic response that was different from just getting saline and just the benefit of getting the surgery, versus when people experience the psychological intervention and support of having a psychedelic trip, the ketamine response for depression lasted for over 6 months. And so there seems to be some difference here. And that's also maybe another important debate that's going on in the field, is whether or not it's important to have an actual trip. So there are some groups that are bioengineering these psychedelic molecules to make it so that they don't have a subjective effect. So you would essentially take it and then you wouldn't experience anything, but maybe it also has some benefit in the brain. And I think that our group tends to lean along the lines of, if you want the long-term if you want the long-term benefits that seem to have a lot of a durable therapeutic response, you need the trip to go through, but it doesn't mean that the psychedelic effect that you might see without the trip, it doesn't mean that the therapeutic response you might see without the trip isn't maybe helpful, especially for groups who maybe can't go through a trip. Like if you have schizophrenia, or if you're just in an older population where you really, really distrust these drugs still, based on a lot of the advertising and like the propaganda that came out about them in the like war on drugs and in the mid-90s. And so for us, trip is extremely important, but that's an active area of debate in the field right now.
Give Makenzie another moment.
Thank you. Woo.
Oh, thanks.
Here you are. Yeah. Make a little clip onto your collar here.
The octopus stuff sounds fascinating.
The octopus stuff is really cool. I wish that a lot more of their screens. This solution that's developed is this one, but I think I'll share more about what the result of
You have them in the lab, you've got tanks in the lab?
Yeah.
How many octopi you got?
I think we're at like between 25 to 30 right now.
It's a lot. Yeah, it's fascinating.
Okay.
Great.
Okay, I'm gonna go ahead and introduce Lauren. So Lauren Sigda from the teaching lab at UC Berkeley. Lauren grew up in New York and has always loved biology. She went to undergrad at the University of Rochester, where she majored in brain and cognitive sciences and art history. After a Fulbright year researching in Vienna, she then started her PhD at UC Berkeley in the Vision Science Department and joined the Teuton Lab. She is currently a third year in the program, and in her free time, she loves to hang out with her friends, listen to music, and exploring new cafes and restaurants.
Yeah, thank you. So thank you all for being here and for the introduction. I'm Lauren, and today I'm going to be talking about what makes you see better. So, okay. We're probably all familiar with a chart like this. This is the Snellen eye chart. It's the same thing you'd see at the optometrist's office. To be clear, this is not a clinical test right now. The distances are all off. But you can try it yourself and see how far down along the chart you can get. I see some people are trying one eye versus the other. But yeah, does anybody want to share? How many of you wear glasses? Probably the vast majority. But yeah, so this is a good test for not only how good our eyesight is in general, but a good resolution of our acuity specifically, which is our attention to fine detail, or basically the resolution of the eye. So the eye works sort of like a camera in that it's a really high quality camera, and we have a lot of sensors in the eye that can pick up details in the environment compared to, let's say, a really lower quality camera with fewer sensors. And because of that, it works in the same way, where we know with cameras, there's a lot of components that limit our ability to resolve things, like the shutter size, shutter speed, other technical terms. So let's talk a bit more about the eye. So let's pretend that this is our eye over here in the corner, and we want to look at this moderately fine quality image of a mountain. So we could gaze out at a branch on the tree, and let's think about what are the stages of processing that our eye is going to go through while we try to process what it is that we're looking at. So first, we're going to pass through the visual optics of the eye, sort of like a sensor. And then this is going to pass to the photoreceptors, which line the back of our eye, which I'll talk about in a minute, which sample the image as the image moves around the cells. And then this is sent to the brain, where it does some really fancy processing to understand, is this a tree? Is this part of the mountain? Is it in front or behind? How important is it for survival? And then ultimately we perceive it's just a branch. So vision ultimately begins in the retina. So everything that we perceive is basically coming from photons, which reflect off of surfaces in the environment. And they travel through the eye to the retina, which is this thin tissue lining the back of the eye here, which is made up of about 130 million cells. So that's really, really dense. And as the light passes through to the back of the eye, we have this literal depression or pit called the fovea, which is our center of best vision. And that's where the vast majority of all our cones and the vast majority of our cell processing is done. These cones are photoreceptors, which allow us to process all of our color vision, all of our high acuity vision, and everything that's important in our daily life, specifically when you're reading, maybe when you're driving. Your fovea is doing the heavy work here. I'll just say, it might seem a little counterintuitive that the fovea is so small in compared to the rest of the retina, but there's a lot of other processing that goes on other than just your bestest vision, like light signals in the dark. There are other cells that handle that. So acuity ultimately depends on where the fovea is in the world. And like I said, all your best vision is at the fovea and everything else is kind of blurred, but we're not conscious of it because of processing that the brain is doing. You can try it yourself. If you're staring out into the world at your hand and you move the hand away from your fixation target, it's going to get really blurry really fast. So ultimately, us with glasses, we know this, the eye's optics are not perfect, but let's pretend that we had emmetropic perfect vision, they still would be wrong. It's sort of like the camera of the eye is looking through clear water in the ideal scenario versus a camera eye, which is looking through rippled water. We have aberrations and things going on through the eye, which limit the light's ability to hit the retina perfectly. So let's test this. Can everybody see this illusion so far? So we have a type of aberration called longitudinal chromatic aberration, which basically means not all of the rays of light of different colors are going to hit the same spot of the retina. So maybe I can get some hands, some gestures. Who sees the red in front of the blue? Okay, solid group. Who sees the blue in front of the red? Okay, also solid. Typically, people would say the red is in front in this case, but it ultimately doesn't matter. It just means that your eye's optics are not the same for the blue and red focal plane. The red is typically focusing a bit further back in the retina, and the blue is a little bit more front. Is everybody here okay with flickering stimuli? Okay, this might help test it out a little bit more in case you wanted to see it the other way around. So you might perceive the depth of the focal plane between the blue and the red changing just as some of the other depth cues of the image are changing. Is that good? Okay. Yes, so not only are the eye's optics an imperfect system, but we also have something called fixational eye movements, which means even when we're trying our very best to fixate on a single spot in the world, we're still drifting that image over our fovea to get as much sampling as we can. So this is called the rotating snakes illusion. You may have noticed by now, there is no video playing. It's just your eye trying to sample as much as it can in a really dense environment full of information. Is this working for everyone? Okay. Okay, so we know that the photoreceptor sampling process is ultimately constantly moving. So is there a way that we can measure that in an optical system? How can we bypass the natural imperfections of the eye in order to achieve better acuity? So, analogy, has anyone ever looked out at the night sky and tried to fixate at a single star? You may have noticed it was kind of twinkling a little bit. There's kind of a halo effect for a really bright star on a really dark background. So something that was developed close to 40 years ago is a process called adaptive optics. And this is something that was used in a telescope to allow us to de-twinkle the stars and get a better view of things in space in general. Now, through this same process, we can de-twinkle the eye and ultimately correct for the aberrations of the eye and also track it with really fine resolution to correct for the movement of the eye. So this is an example of without the process of adaptive optics, and this is what would happen when we turn it on using the same equipment, same everything technique. So how do we do this in the lab? We use something called the adaptive optics scanning laser ophthalmoscope, which both images and tracks the human retina on a cellular scale. This is done basically using a laser beam that is moving really, really fast in really, really tiny increments in order to track the area that we're interested in on the eye. This is an example of somebody sitting in at the system. Any questions about this? We can come back to it later. So here's a great example of something from my research. This is my retina. These are the cells in the back of my eye as I'm fixating as best as I can at a target and performing an acuity task. So you might see, depending on your acuity, that there is an E that's going to appear to a point, and it's oriented in a specific direction, and I, as the observer, am doing my best to report the direction of the E. So we do this basically by dropping out pixels in the laser beam in the shape of the E, and ultimately, we have proven time and time again that using adaptive optics improves people's acuity. So we might know that 20/20 is a pretty good prescription. 20/20 vision is basically standard. With adaptive optics being on, we can drop acuity down to 20/8, which is extraordinarily difficult in normal seeing observers. So other things that we can do using this system are also stabilizing the same target to a patch of the retina. So you can see each little dot here is a foveal cone. We can track and pin the same stimuli down to the single cone resolution. So you can see if the E is stable in the world as we're looking at it now, the eye is moving over it many, many times per second, and creating sort of an average composite smear that we're using to create the image of the E using our brains. But we can also pin it to the retina where there is no dynamic sampling at all. Can anyone guess what would happen in this case if you stabilize something to the exact eye motion that you're naturally doing?
Burn your eye.
No, everything here is perfectly safe in humans. But actually, it would fade from view completely. You would no longer see the E at all. But yeah, so in conclusion, we use in vivo adaptive optics imaging to view structural features of the eye and also track it on a cellular level. Thank you so much for your attention.
talk a bit more about why it disappears from our eye when it's hitting the same spot?
Yes. So the idea is basically when we track stuff with the same motion of the eye, to the exact degree of that motion, we don't have enough information to sample the environment anymore, and it sort of fades from view, probably because we're adapting to it and it's no longer important. But ultimately, it's not known. We just know that it does. There's a lot of theories out there, but my favorite is that you need more information. And over time, because we're not consciously moving our eyes, we're just fixating on the thing, we've decided it's not important.
Is the whole eye moving, or are there like tiny muscles like in
the whole eye moves? The whole eye moves as one. Both eyes move at the same rate as well. So we can't separate that out. But if you could, we could talk. That would be interesting.
Can you explain how the adaptive optics works? Like what is actually going on? Because I think I assumed that you were stabilizing it to the eye before you showed that you can't stabilize it to the eye or it's something just.
Yeah, so I don't have a perfect image of how this works in the presentation, but the idea is that we have natural aberrations of the eye, which, like one example was the longitudinal chromatic aberration, where things are focusing in different planes on the retina. We also have transverse chromatic aberrations, where things in time and space are not lining up exactly on the retina. And so we all have our own personal aberrations, which we adapt to and we don't notice in our daily life. But using adaptive optics, we can take the light that's coming off of the laser and it shoots back out into something called a deformable mirror, which corrects for this in real time. So sort of like when you press on a piano key and it hits that hammer in the back to make the sound, it's a little bit like that. So let's say my aberrations shoot out to the deformable mirror, it's going to correct back the opposite perfect beam so that you perceive no aberrations. Yeah.
What is is there like a clinical application for this, or like something you would hope to use this type of technology for to help correct people's vision?
Yeah, so right now, one of the projects that we're working on is sort of about a disease called retinitis pigmentosa. So the idea is that in retinitis pigmentosa, a lot of your cones drop out over time as part of the chronic inflammation of the disease, I believe. And so we can simulate this same cone dropout in a healthy observer so that we can test different ideas about how many cones you need to see, which informs people who are actually doing physiology work what the thresholds might need to be so that their subjects can see better and things like that. But there's one particular finding that is recent that is not very clinically relevant yet, but it's from last spring. Our lab put out a paper about a color called OLO. So the idea there is that we can target only cone types that respond to medium wavelength cones, or the green color, and create a new color perception that is not naturally able to be seen. So with that, we have proven that we can simulate single cones and create new perceptions of things. So the idea is there's a lot that can come from it that we haven't even thought of yet, but the technique is sound. Yeah. Any other questions? Yeah.
Did you get to see the new color?
No, I did not. I did not want to sit for the experiment for a long time, which is how that would go. There were only 6 people that saw it, which is half the lab, I guess. But
Some of my friends were really excited about it.
has anyone done oh no, not since the experiment was finished, but there are similar things that are undergoing experimentation at the moment. Yeah.
Could you do the same thing but test like the short or the long wavelength and bring the color that way? Did you just happen to choose
medium.
Yeah, the medium cones have the spectral wavelengths which are more unique compared to the short and the long, which have more overlap. So the M range was better evidence to prove that nothing else is being stimulated, and only the M cones could have possibly been eliciting this color sensation.
How was
Yeah.
that color described?
Kind of a teal. like an interdimensional teal, if you will. I haven't seen it. I've only heard rumors. But yeah.
See through this experiment, or could it be
Yeah.
replicated kind of like physically or something?
You can replicate something similar to it in a way that I wouldn't recommend. Theoretically, you could stare at a red light for a really, really, really long time and then look at something green, but it wouldn't really come close to this idea. And please don't do it. Yeah.
I have a question about the visual acuity graph that you're showing here. So you're saying with this technique, there's an increased acuity. Was this tested with the eye chart that you showed at the beginning, or was it more like images?
Yeah. So typically when we do experiments in the AO, because the resolution is so fine that even small head movements would cause a disturbance between the imaging of the system and the aberration correction. So we have people who are observing an experiment on a bite bar, which for those of you who don't know what it means your head is locked into place and they can't respond with what they're seeing. So in these experiments, we typically show an E, just like this, a Snellen acuity chart E, and have it oriented in 4 different directions, which in my experiments correspond to the 4 buttons on an Xbox controller. So I believe that's what was happening here. Yeah. Yeah.
Can you explain what 28 vision would be? Does that mean you could see a blade of grass? From
Yeah.
a distance, or what would that enable?
Sure, basically. But the idea of acuity is, so at 20/20 vision, you can see at 20 feet what the vast majority of other people can also see at 20 feet. So let's say for 20/8 vision, you could see at 20 feet what most people see at 8 feet. Yeah. Okay. Thank you. Are there Zoom questions?
No Zoom questions.
Okay, awesome. Thank you.
If you've enjoyed hearing these talks and are not currently set up to hear more talks in the future, you can join our listserv using the QR code that Kaylin is about to put up. Or you can also follow our Facebook or their social media account. We will also upload the seminar to our YouTube page where you can find our archives from past seminars. And we will also send out a post-seminar email with resources from our speakers. We can follow up with more information. We will be back next month on April 16th to talk about research. And this research will be focused on the aging brain as well as robotics for radiation testing. Until then, we'd like to thank our speakers for their talks one last time, as well as you all, our audience, for joining. Thank you.
This is the diaphragm or the fluid? Oh. I think this is the diaphragm, right?
Yeah.
They were sitting on.
We're going on a little bit of a tour, and I think this is actually going to be our very last stop in the tour, where we're actually going to be at the South Branch, or the March 17th one, not the Central Branch. You can sign up for our listserv, and we'll be sure to tell you exactly where we're going to be there. So I don't think that we'll be here next month.
Yeah.
We're gonna do a tour where the sponsors are.
Sure.